Hello, good morning and welcome to our session. Today we'll be discussing chapter number 21, nervous control and coordination. Okay, we'll be directly going to the first question. Identify A, B, C, D respectively in the graph below. This graph is not in our books. This is a graph that we can mark based upon the observation we make in a dual beam cathode ray oscilloscope. See, during a active transportation of impulse across an axon, you can insert the two electrodes of an oscillon. Remember, it is an excitatory electrical current that is passing along the membrane of an axon. That is what's called impulse. Impulse is just an electrical pulse. This is flowing through the membrane. So, you insert an electrode to the inside and another one to the outside. And what is the electrical potential that is developing there during this process? You can measure not by a voltmeter, because voltmeter will be used for measuring large quantity of electrical disturbances. Here it is only very slight, and it is in millivolt. 1 by 1000 of a volt is 1 millivolt. This can be measured only by a dual beam cathode ray oscilloscope. Okay, so we are giving to the amplifier, and from the amplifier it is given to the cathode ray electrode. So what will be the nature obtained? We will be having the values recorded and the changes recorded in this way. We know that when a axon is not conducting any impulse, membrane is at rest, there is a potential developing. That's called resting potential. During that resting potential, the value may be varying from minus 40 to minus 90. We take the average of that. That's why we say that resting potential is minus 70. Why we say it is minus 70? Why we say that it is minus 70? Inside is negative. That's why we take the value negative. Negative means inside is negatively charged. Okay. This is the average value. Okay. So resting 70. This is the 0. And from here you will be having plus values. This is the minus values. This is the neutral. Okay. And we have to apply a potential or a stimulus which is sufficiently strong enough to activate a neuron. That minimum strength required for activating a neuron is called threshold value. So it should be minimum 10 to 15 millivolt higher than that of the resting potential. Resting potential is minus 70. So this should be at least 10 to 15 means 10, minus 10 higher. That means it should be minus 60 or minus 55. So minimum minus 60 we have to give. That minus 60 is the threshold. Okay. So we are having, this is the resting potential. So resting potential is 70 and this is minus 55 to 60. Then only a neuron become active. And because of the influx of sodium, inner side is becoming positive and it goes past that 0 and then goes to plus 30 millivolt. That plus 30 is the action potential. So resting potential is minus 70 millivolt plus 30 is the action potential. Then again it come back because the sodium ion channels are immediately closed and potassium is again moving out, and because of that there will be again coming back. That is coming back to polarized condition is called repolarization. So B represents depolarization and C represents repolarization. And during repolarization, remember, the entire process will be completed in a very small time period of 0.5, in a small period of 5 millisecond. Okay, it is so quick. And for a fraction of a millisecond, 0.5 millisecond, that is 0.5 millisecond, the potassium ion channels are more widely open. That means more potassium is going out. As more potassium is going out, the inner side is becoming more negative. The value goes even past minus 70 and it may reach even minus 80. Now, immediately the potassium ion channel regain the original permeability, and now it is coming back to minus 70. These are all measurable using double beam, dual beam, oscilloscope. Okay. This is the graphical representation of all the activities that are all going on in an axon. Okay. This is not in our books. So, A, we are having the threshold stimulus applied. Okay. This threshold stimulus will be minimum minus 55 to minus 60 millivolt. Now, B is depolarization, C is repolarization, and D is hyperpolarization. It is called hyperpolarization. Why? Because inner side is becoming more negative. So the option goes to apple. Which of the following is the correct difference between rod and cones of the retina? See, more concentrated in center of retina. See, which is the correct one? Rods are more distributed throughout, whereas cones are concentrated. So this is not a correct one. Visual acuity. Rods are having low. They are sensitive to low light, and acuity, clarity is also low. For cones, it is very high. Visual pigment content is in rods rhodopsin. It is not iodopsin. Here it is. In cone cells, iodopsin. Iodopsin is called visual. Actually, cones are having this iodopsin, visual violet. Rods are containing rhodopsin, visual purple. Overall function, vision in poor light. That's why it is called scotopic, black and white vision. And color vision, detailed vision in bright light. That's why it is called photopic. That is the only correct one. Others are all mutually changed. The wrong statement regarding the human brain. The three layers of meninges from outer to inner layers. What are they? Outer one is the dura mater, outer, which is strong mother. The middle one is the spider web, arachnoid, and inner is pia mater, soft mother. That is correct. So the three layers of meninges from outer to inner. The outermost one is dura mater, not pia. This is the innermost. Okay. Middle one is arachnoid. Cerebrum, thalamus, and hypothalamus belong to the forebrain. Correct. Corpus callosum connect the two cerebral space. That is characteristic of mammals. Corpus callosum connecting the right and the left hemispheres. Gray matter indicates the presence of neurons. That is why they are all gray. White matter is mainly because of the myelin sheath of the axons. So they are all containing the axon covered by myelin sheath. That is why they are white in color, white matter. Association areas of cerebrum mainly control memory and communication. Correct. So only the wrong one is the first one. Incorrect match. Diencephalon, it is correctly called thalamencephalon. See, brain consists of three parts: prosencephalon, mesencephalon, rhombencephalon, forebrain, midbrain, and hindbrain. Telencephalon is for amygdala. Mesencephalon is midbrain, correct. Myelencephalon, medulla, and metencephalon for cerebellum. These are all correct. Okay. So, diencephalon is thalamencephalon. Pons is actually a part of the hindbrain associated with the cerebellum. Pons veroli, that's the full name of that part. Fovea. What is fovea? Fovea centralis. It is the central point of the macula lutea, thinned out part. It lies at the center of the macula lutea. Macula lutea is called yellow spot. It is devoid of blood vessels. There will be no blood vessels and nerve fibers. Okay. Region where only rods are. No. This is the region where maximum acuity, maximum resolution, most clarity. Pictures are seen when images are falling on this part. So they are all having only cones, not rods. So that is why it is wrong. Region of greatest resolution, clarity. Region where the ratio of photoreceptor, bipolar, and ganglia cells. These are the three layers of cells forming the retina, and this is the ratio in which these cells, the outermost photoreceptor, middle bipolar, and ganglia cells are in the ratio 1:1:1. So only 3 is the incorrect one. Pinna. So what is pinna? Correct vibration in the air which produce of the sound. It is characteristic of mammals, but aquatic mammals are having as an adaptation for swimming life. They don't have this pinna. Then tympanum, eardrum. What is the function of the eardrum? Eardrum is increase the efficiency, passage sound, transfer sound waves to the ear ossicles. Okay, so we have arrived at the answer now because A is 1, B is 5. Immediately we came to the answer. Now, continue answering. Auditory meters. Meters means the canal. We are having an auditory meters. What is that? Auditory meters. Auditory meters is the passage of sound wave from pinna to the eardrum. From pinna, the sound vibrations are brought to the eardrum through this canal. Auditory meters. Ear ossicles. We are having 3 ear ossicles that increase the efficiency of transmission because they act as a leverage system and will be also increasing magnifying the sound vibrations to about 10 times. So our middle ear is a built-in amplifier. It is not only conducting sound vibrations but also acting as an amplifier, built-in amplifier. 10 times amplification is done because of that leverage system and 2.2 times. So 2.2 times because of the ratio of area between eardrum and oval window. The pressure in a larger area when transmitted to the oval window, the ratio of surface area is 2.2. So it will be magnified, strengthened 2.2. By the leverage system, 10 times. So total 22 times. The sound vibrations are amplified to a maximum of 22 times because of our middle ear. That is why I told you it is a built-in amplifier also. Cochlea. What is that cochlea? Cochlea has hearing receptors, organ of Corti, and eustachian tube. Eustachian tube equalizes the pressure on either side of the eardrum. See, the eardrum, tympanum should be maintained always in a fully stretched condition. It should not be, okay, it should not be pushed either to this side or to this side. It should be remaining in a fully stretched one so that the pressure on either side of the tympanum should be always same. And Eustachian tube is keeping that pressure on either side equal. Okay, so option goes to cat. Group of structures involved in maintenance of equilibrium. We are having the ear having two parts or two regions. The lower cochlea is concerned with hearing and upper vestibular organ. Vestibular organ is concerned with equilibrium maintenance. That's why our ear is statoacoustic. The equilibrium and hearing. Vestibular organ consists of three semicircular canals which will be having crista ambularis, a swelling called ambulus present within which there is a sensory organ called crista ambularis. So semicircular canals and the otolith organ. This otolith organ consists of upper utricle and lower saccule. Present in the saccule and utricle, you have another structure called macula. So, crista present in semicircular canals are responsible for equilibrium maintenance when our body is tilting to any angle. That is called angular equilibrium or dynamic equilibrium. Whereas this macula is responsible for static equilibrium when you are not moving or when you are moving only in a straight line. That is called linear equilibrium. So macula is bringing about static or linear equilibrium, whereas crista in semicircular canals are responsible for maintaining the angular and dynamic equilibrium. So semicircular canals, utricle and saccule. Okay. Cochlea is concerned with only hearing. Action potential through an axonal membrane is represented. Which of the following is true? See, potassium is normally moving out and sodium is not at all entering in. But when you apply a stimulus, here you can see that normally we are having positive outside and negative inside because we are having already lot of sodium outside plus potassium is also moving out. So outside is positive. Now when a stimulus is applied, sodium ion channel immediately is open and sodium comes in. So that potassium outflux is reduced. So sodium is coming in, potassium outflow is reduced. So accumulation of positive charges. So the polarity is reversed. This is called depolarization. Immediately there will be an induction in the neighboring point. The next point is induced to depolarized, and by that time the original point become repolarized. So this point where the stimulus applied is immediately depolarized and again repolarized, and the next point is now depolarized, so that how it reaches, there will be a flow of charges. This is the polarized and this is the depolarized. Okay, outside is negative, that's why it is depolarized. This is polarized, outside is positive. Charges will flow from positive to negative. So from the polarized to depolarized on the outside, and on the inside from depolarized to polarized. That's the clue. Okay. A current flows on the inner surface from site A to site B. On the outer surface, current flows from site B to site A to complete the circuit. Correct. See, always charges flow from positive to negative. The polarity of the site is reversed and the action potential is generated. Now, action potential is generated in site A. Depolarization followed by repolarization. It's a cyclical event. Okay. That cyclical event is called action potential. So, action potential is generated in A, that will flow to the next one, to the next one. So, in the form of a wave, it passes along the membrane. Impulse generated at site A arrives at the site B because of the flow of charges. That flow of charge, that's called local current. That local current is flowing, which is called impulse. This is why we say that impulse is a wave of, it is an excitatory wave passing along the membrane of an axon. Why I call it as excitatory wave? That wave is created only when a neuron is excited, activated by a stimulus. A neuron cannot produce a signal by its own. Only when it is excited, when it is activated, then only it will be. This is called excitatory electrical wave passing along the wall of a activated neuron is called impulse. So all of them are correct except for the third one. Regarding electrical synapses, synapses are points where there is contact between a neuron and a muscle or between two neurons. Here it is electrical synapses. The membrane of pre and post synaptic neurons are very close. The cleft will be so small, 0.2 nanometer, correct. Transmission of impulse across an electrical synapse is similar to the impulse transmitted along an axon because there is no need of a chemical. There is no involvement of a neurotransmitter. Impulse transmission across an electrical synapse is always faster because there is no need of the vesicles to come fuse, release of the acetylcholine or the neurotransmitter, etc., etc. They are all rare. We are having very few such electrical synapses. We are mostly having chemical synapses. Okay. These electrical synapses are abundant in invertebrates. They are all first observed in abdominal nerves of crayfishes. So, all of them are true. The gap is very, very small. Again, there will be a question coming in connection with this part. I will explain again there. The three concentric layers formed in the eye. What are the three layers in the eye? Outer is the sclera. Sclera is the hard outer. Then choroid. Choroid means vascular. And retina. The innermost is the retina. So, external layer is the fibrous, middle is highly vascular, inner layer is the nervous cord. Same one. So, it is the retina. Sclera, choroid and retina. So, option goes to apple. Incorrect difference between the myelinated and non-myelinated. What is myelinated axons? An axon where around the axon, there may be some glia cells. These glia cells may be Schwann cell in peripheral nervous system or oligodendrocytes in central nervous system. These are the two types of glia cells. Understand that glia cells are constituting more than 50% of the total nervous tissue. They are very important. We can classify glia cells into microglia and macroglia. Okay. Macroglia are large. Microglia are small. Microglia are mainly macrophagic, amoeboid, whereas macroglia are all having different functions, covering, protecting, helping in the conduction of impulses, helping in maintaining a climatic environment for the normal physiology of the neurons, etc., etc. We are having a large number of different types of cells like astrocytes, oligodendrocytes, Schwann cells, radial cells, stellate cells, etc. And this is the axon. You are having the Schwann cell around it. This is the nucleus of that. So, if I take a cross section, I can show it like this. Axon membrane, and around that you are having the Schwann cell, sometimes coiling in concentric rings. These concentric rings will be thus insulating the axon in the form of a thick layer which is very rich in sphingomyelin, a specialized phospholipid called sphingomyelin. So, since it is forming a thick layer, insulating layer, such axons are called myelinated. So, this is one cell, next Schwann cell. Schwann cells are arranged end to end, one after another. And sometimes they may be forming concentric coiling around it. Okay. So that it will be forming a thick layer, insulating layer. In myelinated, myelin sheath is present. That is the reason why it is called myelinated. Myelin sheath is absent. Okay, that's the correct statement. Schwann cells are present in myelinated and it is absent is that part is wrong. Schwann cell will be present, but that Schwann cell is not forming concentric covering, concentric coiling. Schwann cell is present in both myelinated and non-myelinated, but it is not forming that concentric coiling around it. Schwann cell is absent is wrong statement. Seen in myelinated is usually seen in central nervous system. Non-myelinated are seen in the autonomous nervous system and sympathetic system. They are not present in, see, it is just the wrong way. It is present in the central nervous system, spinal cord, cranial nerves. In autonomous and somatic nervous system, we are having non-myelinated. It is saltatory conduction. What is saltatory conduction? The impulses are jumping from one node. Say, for example, this is the node. What is the node? A junction, the point where there is no myelin sheath. That narrow gap in between adjacent Schwann cell. This is called a node. The ionic channels are much concentrated here that the voltage produced here will be so powerful that it can jump from this one to this one. In the region in between, it's called internode. This is the node of Ranvier. Okay. The ion channels are concentrated in the nodes so that the action potential generated is so powerful that it can jump from one node to the next node. So, the electrical charges are jumping, not flowing. Okay. It is flowing in a non-myelinated. That's why it is slow. Whereas it is jumping and so will be very fast in myelinated. This is called saltatory conduction. Saltation means jumping. Whereas in non-myelinated, it is point-to-point transmission. It is flowing. Positive, negative, point-to-point flow. Whereas in myelinated, it is jumping. Neurons with one axon and two or more dendrites. This is called poly. Okay, we call it as multipolar. This is a multipolar. Present in the cerebral cortex, neurons with one axon and one dendron. Only two processes found in the retina. This is called bipolar. Okay, this is multipolar. Neurons with non-polar process. This is apolar. It is seen only in embryonal cells. So, what is order? Multipolar, bipolar, apolar. Multipolar, bipolar, non-polar, also called apolar. Apolar. Missing word. Depolarization of nerve cells involved. See, this is an axon. In rest, no sodium is coming, only potassium is going out. Membrane is freely permeable to potassium and nearly impermeable to sodium. So, that sodium is coming in and potassium permeability is reduced. That influx of sodium is responsible for depolarization. Involves influx of sodium, coming in of sodium, and repolarization during nerve impulse conduction is due to outflux of potassium, exodus of potassium ions. Okay, so all the incoming sodium are pumped by sodium potassium pump, and potassium will be increasing its permeability. It resumes its original permeability, so potassium will be moving out. That's how it is again coming to the normal polarized condition. So exodus of potassium. False statement. Axonal membrane is comparatively more impermeable to potassium. Okay, we can see that axonal membrane is more impermeable, not more permeable to potassium, and almost impermeable to sodium. Okay, so that one is a wrong one. Membrane is impermeable to negatively charged protein. Correct. That is why the amino acids present inside is not moving out. That is why the inside is negative. Axoplasm inside the axon contains high concentration of potassium and negatively charged protein and low concentration of sodium. Fluid, you can see that. This is the condition. Okay. Sodium is almost 10 times outside and potassium 30 to 40 times inside. So, from a high concentration to a low concentration, potassium is moving. So, that potassium outflux is causing outside positive. Fluid outside the axon contains a concentration of potassium, a low concentration of sodium, and forms a concentration gradient. It is containing a low concentration of potassium. Potassium 30 to 40% inside than outside. So that is also wrong. So first and the last one are false, the others are true. 2 and 3 are true. Limbic system, part of the brain. Okay. And hypothalamus do not. See, limbic system is having sexual behavior. Limbic system is having amygdala, hippocampus, and septum. And that septum is responsible for sexual behavior, emotional reactions, amygdala, anger and fear, also because of amygdala. It is the motivational center. And hippocampus is concerned with the long-term memory, smell, etc. Whereas vision is the occipital lobe of the cerebral hemisphere. So it is not part of the limbic system. Septum, amygdala, and hippocampus are the three parts of the limbic system. Lip-like structure. Which of the following is the correct representation of reflex arc? In a reflex arc, there will be a receptor. We are having a diagram. I will explain it again. A reflex arc consists of a receptor organ for receiving the senses. Then it is taken by a sensory to the central nervous system, which is spinal cord or the brain as the case may be, through a afferent neuron. Now, in the central nervous system, there will be an interneuron connecting them, and orders are given by the central nervous system through a motor. Motor is also called efferent, that bring the command to a motor organ, motor end plate. This is the route taken by the impulse in bringing about a reflex action. This is called reflex arc. What are the neural elements that are involved in bringing about a reflex action is called the reflex arc. So you should have a receptor, then a sensory, that should be the order. A central nervous system which contains the interneuron, a motor neuron, and an effector organ. Motor organ is the organ where the effect is done. That is why it is called effector organ. Okay, so that is the correct answer. In a knee-jerk reflex action, the spindles are the sensors and muscles are the effectors. Order of the 3 layers of cells in the retina. We are having from inside to outside. We are having the ganglia cells. Okay. Then bipolar cells. Then rods and cones. That is photosensitive. So light is first touching the ganglia, then bipolar, then the rods and cones. Ganglia cells, bipolar cells, photoreceptor cells. This is the 3 layers. And remember, our retina is a inverted retina. The arrangement of cell layers are just opposite to what we normally think of. That's why our retina is said to be inverted retina. Normally we believe that light will be first coming and hitting the rod and cone, photosensitive. No, our retina is just like that. It is on the farthest, okay, outermost layer. So the light will be first touching the ganglia cells, then bipolar, and then rods and cones. Clear? The function of the ear ossicle, I have told you. Ear ossicles are amplifying, not only conducting, but also amplifying, increasing the efficiency. That is amplification of the sound vibrations. Equalizing the pressure on either side, eustachian tube. Maintaining the equilibrium of the body, it is the function of vestibular organs. Increasing the efficiency of transmission is the function of the ear ossicles. It is not only simply conducting, but also amplifying. Enclosed in the membranous labyrinth, that is the bony labyrinth. Collecting the vibrations in the ear, that is the function of pinna. We have studied this question earlier. Now, there is no correct option given here. Why? I will explain. Organ of Corti is a part of the cochlea and located on the basilar membrane. So we can better take located on the basilar membrane. There is no other option. So it should be 4. Okay. What is cochlea? Cochlea is the coiled part. Again we take it. Eustachian tube, there is no answer for that. Eustachian tube is not attached to the cochlea. It is not located to the basilar membrane. Okay, there is no other. It is not attached to the oval window. So there is no correct option for that. Stapes. For that we are having stapes is the innermost one ear ossicle attached to the oval window. So this one. If you take this one as the correct answer, organ of Corti, a part of cochlea. Okay, but what is cochlea? Cochlea, coiled part of the labyrinth. Okay, for this one also. And eustachian tube, again there is no answer. Located on the basilar membrane. So both 1 and 4 are not matching for this C. Eustachian tube is a tube connecting the middle ear with that of pharynx, mainly responsible for maintaining the pressure, equalizing the pressure on either side of the tympanum. So that efficiency of hearing is, so that should be neither it is a part of cochlea nor located in the basilar membrane. So this is the problem. So the more correct answer can be the second one. In mammalian eye, the fovea is the central point, is the central point of the visual field, where it is the central point of the macula, lutea, yellow spot, where we are having only cones, more rods, no high density of cones occur, correct? There will be no rods at all, only cones, high acuity, optic nerve leave the eye, that is the blind spot. For example, this is the fovea centralis part, which is located in the central part of the macula, lutea, yellow spot. Just below that, you have the blind spot. It is the blind spot where there are no rods and cones. That's why it is blind. Optic nerves are leaving the retina and blood vessels are entering. Okay. Only rods are present, no rods. So, the correct answer is 2. Destruction of the anterior horn. It is not in our books. See, this is the spinal cord. From spinal cord, spinal nerves are coming. And each spinal nerve will be arising from 2 roots. A dorsal root and a ventral root. Remember, all our spinal nerves are mixed. You have an afferent, that is sensory, and a different part. Okay. Now, in spinal cord, gray matter is inside. That gray matter is extending in the form of 2 extensions. That's why it is called gray horn. So we can simply say that the structure of the spinal cord is almost like a letter H. It is almost like a letter H. Okay. These are the 2 dorsal horns and these are 2 ventral horns. Dorsal horns are posterior and ventral horns are anterior. Sensory nerves are connected to the dorsal horn and motor nerves arise from the ventral. Clear? Here, there is one-way traffic. All the informations coming to the central nervous system are coming through the dorsal route. They will be first coming to the dorsal horn. That is the posterior. Now the question is, what will happen if the anterior horn cells, this is the anterior horn, they are all damaged. When they are all damaged, no information is coming. Motor impulses are coming. Remember, this is the root of motor neurons. Different or motor neurons are arising from the ventral or anterior horn. When the cells of this part is damaged, no motor impulses are coming. Voluntary motor impulses will not be coming. Okay, this is voluntary because autonomous will be coming. This is involuntary. Okay, but this is voluntary. Voluntary motor impulses will not be coming. This is the structure showing the spinal cord and the origin of the spinal nerves. Each spinal nerve is arising from 2 roots, dorsal root and ventral root. You understand the linings? Impulses coming through the dorsal horn only and leaving through the ventral horn. Ventral is anterior. And choose the correct statement. Photoreceptors receiving light in the human eye are depolarized during darkness and become hyperpolarized response to. That is the action. By our retina is sensitive to light. When light fall on it, there will be a breaching. Okay. There will be a signal generated. That is the ion channels. Sodium ion channels are closed so that when the sodium ion channels are closed, the cells become hyperpolarized. That acts as the signal. This signal is transmitted through the bipolar to the ganglia cells, and ganglia cells will generate action potential. That's how a signal is generated in the retina and is conducted through the optic nerve to the brain. Okay. That's correct. Receptors do not produce graded potential. Receptors do not produce graded potential. They produce graded potential. See, what is the stimulus? Amount of stimulus. If you apply a small stimulus, okay, receptors do not produce graded potentials. Potentials will be differing. No C receptors. No C means harming. There are pain receptors respond to the changes in pressure. No. Mason's corpuscles are thermoreceptors. See, thermoreceptors are caloreceptors and frigidoreceptors. Caloreceptors are responsible for recording the heat. It is referees corpuscles. Frigidoreceptors are receiving cold. Cross corpuscles. See, there is a law called all or none law. In impulse conduction. So, the correct one is photoreceptors. See, receptors do not produce graded potential. Receptors produce graded potential. Okay. This is the correct one. Mason's. What are Mason's corpuscles? Mason's corpuscles are sensitive to touch or pressure. Touch or pressure. Okay. Then myelin sheath is produced by, who is produces myelin sheath? It is produced by the Schwann cells. See, we are having, this one is incorrect. Schwann cells in peripheral nervous system, and the same is produced in central nervous system by oligodendrocytes. Both of them are macroglia cells. Macroglia cells. Okay, Schwann cells in peripheral nervous system and oligodendrocytes in central nervous system is producing the Schwann sheath. The answer is 1, not this second one. Astrocytes are acting as blood-brain barrier. Astrocytes are large macrophages, the largest Schwann cells, the largest of the glia cells. Osteoclast cells are not at all a nerve cell or a glia cell. It is the cells present in the bone which is responsible for resorption of, removal of the calcium. And osteoclast and osteoblast are opposite in action. Okay, so never osteoclast. Yes, this was the diagram I was telling you. Receptor site for neurotransmitter. See, this is the axon terminal. We are having a axon end bulb, and this is the dendron of the next neuron. So this lies just anterior to the synaptic cleft. So it is called presynaptic, and this membrane is the postsynaptic. So where are the receptors? Receptors are present in postsynaptic membrane. Okay, in simple diagram, I can show like this. This is the axon, synaptic knob, and this is the dendron of the next one. This is the synaptic cleft. Okay, so this is the pre-synaptic membrane, and this is the post-synaptic membrane. Only in the post-synaptic membrane you are having receptors. That is why the signal will be passing only from axon to dendron. Remember, synapses are unidirectional, sending signals only in one direction. It is because of this type of arrangement. Synaptic vesicles are present only here, and the messenger molecules are falling here. So only from axon to dendron messages are coming. Good vision depends upon adequate intake of carotene-rich food. Select the best option from the following statements. See, vitamin A derivatives are formed from carotene, correct? Vitamin A, carotene. The Provitamin A. Carotene is a provitamin A. Photopigments are embedded in the membrane disc on the inner segment. No, it is on the outer segment. I will give you a small diagram which is not at all in ordinary books. This is the structure of a rod. Okay. In the rod, we are having two parts. Inner segment. Okay. And this is the outer segment. In the inner segment, you will be having Golgi complex, mitochondria, etc. This is mitochondria, abundance of mitochondria, Golgi complex for the secretions, etc. All are present, endoplasmic reticulum, etc. are present here. Here we will be having the nucleus. And in the outer segment, you will be having a number of discs. These are called discs. Discs are present in the outer segment. Okay. This is the structure of a rod. In the disc, you are having the photoreceptors embedded within the membrane. In this membrane, you are having the photopigment. What is the photopigment? In rods, rhodopsin. We are having rhodopsin, visual purple. Remember, they are transmembrane proteins. Proteins present within the membrane of these discs. They are all present on the outer. The cones are also similar to that one, but only in the difference is, okay. In the shape, okay, here you are having the nucleus. There will be slight difference. It is a bit smaller and somewhat conical also. This is rod shaped and elongated. So rods and cones are having this in the outer. Normally this level is not in +2 levels. Okay. That's why you may be finding it a bit difficult in answering this question. Okay. So they are embedded in the membrane of the outer segment, not inner. Is that clear? Retinol is a derivative of vitamin. It is an aldehyde of vitamin A. Retinol is light absorbing part of the all visual photopigments. Correct. So 1, 3 and 4 are correct. So this is the answer. Clear? Transparent lens in the human eye is held in its place. How? The lens is held by suspensory ligament, and that suspensory ligament is attached to the ciliary body. See, we are having the lens kept in position, and this is attached to the ciliary muscle, ciliary body by the suspensory ligaments attached to the ciliary body. And because of the contraction and regulation of the ciliary muscles, the ligament will be pulled or pushed, and it will be exerting differential pressure so that the curvature of the lens will be varying. That is why we are able to see close by and far distant objects. Okay, so it is because of the accommodation, that power is called accommodation, that we are all able to see close by and far away objects. Ligaments are attached to the ciliary body. This is the suspensory ligament. And this is the ciliary body. Ciliary body is the modified anterior part of the choroid, middle layer, choroid layer. Which of the following structure of the regions is incorrectly paired with its function? Hypothalamus, production of releasing hormone, regulating temperature. This is the thermostat of our body. So that's the correct answer. Limbic system. We have studied the structure of the limbic system. It is concerned with smell and long-term memory. Consists of fiber tracts that interconnect different regions of the brain, controls movements. That is pons. Pons means bridge. Medulla oblongata controls respiration and cardiovascular reflexes. Correct. Corpus callosum, characteristic of mammals, bands of fiber connecting the right and left cerebral hemispheres. Also correct. So limbic system is constituted by amygdala, hippocampus, and septum. Okay, in one of the earlier questions, we have discussed it in detail. Now, what are Nissal's bodies? Nissal's bodies are structures which are all present in the cell body, okay, and in the dendrons. They are absent from axon and axon helix. What are they? They were all discovered by Franz Nissl. That's why it's called Nissal's granules. There are free ribosomes and rough type of endoplasmic reticulum. So abundant of rough type of endoplasmic reticulum, that is endoplasmic reticulum with ribosome on the surface, and also free ribosomes. Okay, they are the centers where protein are synthesized. Which of the following statement is correct? Cornea consists of dense connective tissue of elastin and can repair itself. It is not a dense connective tissue. It is a transparent one. That is why light is entering into. Cornea is convex, transparent layer, which is highly vascularized. No, it is avascularized, having no blood supply. That is why you can take the cornea of any person and transfer to anybody else. We simply call it as eye donation. Only the cornea is taken, not the entire eye is taken. Don't think that your entire eye is taken will be donated. No, only the cornea after death. Okay, so that it can be donated to anybody. Why? Because there is no blood supply. So there will be no incompatibility, no immunological reactions. So it is avascular. Cornea consists of dense matrix of collagen and is most sensitive portion of the eye. No, sensitive portion of the eye is retina. Cornea is an external, correct, transparent, correct, protective, proteinaceous. That's the correct one. Covering of the eyeball. And its main function is collecting eye rays to, sun rays to, or light rays to our eyes. Okay, converging rays. And coming to the last question of this chapter, which part of the brain is responsible for the thermoregulator, that is thermostat? Hypothalamus is the thermostat, which is responsible for, see, the hypothalamus is only a 4-gram weighing structure lying on the floor, but is very, very important. It is having a number of functions: controlling thirst, controlling hunger, and also neurosecretory cells. So it is hypothalamus which is responsible for the temperature regulator, thermostat of our body is hypothalamus. Okay, so with that, I come to the conclusion of discussion of the most probable questions from this chapter. I hope you have understood the main concepts. Almost all concepts from this chapter is covered with these 30 questions. Okay. So thank you all. We will meet with another set of questions next day. Thank you. Thank you very much.